This is part 2 of the lecture on viruses. Okay, so we did say that all viruses have a nucleic acid core. It can be single-stranded DNA, single-stranded RNA, double-stranded DNA, or double-stranded RNA. So there's really 4 different types. So in DNA viruses, the DNA directs the host cell's replication proteins to synthesize new copies of that genome and to transcribe and translate that genome into viral proteins. So basically like this, the DNA codes for messenger RNA, and then the messenger RNA codes for protein. Codes for all of these specific amino acids that build the protein. So DNA, the genes in DNA, in all of our DNA, the individual genes code for specific proteins. But first, the DNA has to make a copy of messenger RNA. That's called transcription. So this first part is transcription. So that is what is meant by new copies of the viral genome and to transcribe. And then translation is the second part, when the messenger RNA actually codes and is used to build a protein of a specific sequence of amino acids. So transcribing and translating that genome into proteins. RNA viruses contain only RNA. So RNA viruses can encode enzymes that can replicate RNA into DNA, which cannot be done by the host cell. These RNA polymerase enzymes are more likely to make copying errors than DNA polymerases and often make mistakes during transcription. And so RNA viruses tend to mutate. They tend to mutate faster. And what we're going to learn in a little bit, some RNA viruses, the RNA in their core actually is the messenger RNA. So they skip the transcription part altogether, and the messenger RNA is just directly used to code for the amino acids in the protein. But that's only in one situation. Most of the time, the RNA gets converted into DNA, and then transcription and translation proceed from there. But RNA viruses, just off of this page, the difference in RNA and DNA viruses is that RNA viruses tend to mutate at a lot higher frequency. So ask yourself, the influenza or the flu virus, is that an RNA or a DNA virus? And you should know it's an RNA virus because it mutates so rapidly that we must develop a new vaccine every year for the flu. And then, of course, the COVID-19, the virus that causes COVID-19, is definitely an RNA virus as well because it mutates. We don't know how rapidly yet, but we do know that it's an RNA virus. Okay, so viral receptors. So this is an example of, it says KSHV virion. So this virus, this single virus, which we call a virion, is Kaposi sarcoma. So it's the one that leads to the dark areas of skin on AIDS patients. Let me stop and look up the whole term. Okay, I'm sorry for that interruption, but I forgot how to spell this. It's Kaposi's. Sorry, put my pen back on here. Here we go. So the K stands for Kaposi's. sarcoma-associated herpes virus. Okay, so Kaposi's sarcoma-associated herpes virus is the name of this virus here. And what you can see is there is a nucleic acid core there, and then there is a protein capsid, which is a hexagon shape in this case. And then outside there's an envelope. And this envelope is a phospholipid bilayer with embedded proteins. One of these is the viral receptor. So if you look on the left at the notes, many viruses use some sort of glycoprotein to attach to their host cells via molecules on the cell called viral receptors. This is a little bit confusing because the green molecules that you see, the green oval molecules on the viruses, are viral receptors that belong to the virus, that allow the virus to attach to the host cell. This cell is actually a helper T cell. Which is just a type of immune system cell. It's a white blood cell. And it also contains a viral receptor called the XCT receptor. And this receptor is what binds to the viral receptor in the virus. So they bind to each other, and then you also have CD98 and alpha-3 beta-1, which attach the virus to the host cell. They're sort of like making the attachment more stronger. Okay, so this virus is shaped like a hexagon, but it does contain an envelope on the outside, so it's actually spherical in shape. This image is an image of a bacteriophage. Bacteriophage has a head, which is this part. Inside that head, it has the DNA, has a DNA core, and then the tail is everything below this line. This is the one that looks kind of like a spaceship docking on the moon or something like that, you know, and its shape is considered complex. Bacteriophages are viruses that specifically target bacteria. And you can see this is a head and tail complex shape that you're looking at. So bacteriophage T4 specifically targets E. coli bacteria. Another shape of a virus is the shape of the adenovirus, which does not have an envelope. It does have a capsid, and then these little individual light brown ovals here are capsomeres. So it's a capsid made up of capsomeres. Many, many capsomeres make up the capsid. The capsid is the entire collection of capsomeres. And it also contains these glycoprotein spikes that help it to attach to the host cell. Adenovirus is a double-stranded DNA virus. The shape is called icosahedral. The virus shown clustered here in the picture on the right is transmitted orally and causes a variety of illnesses in vertebrates, including human, eye, and respiratory infections. So we call this icosahedral. HIV, human immunodeficiency virus, the one that causes AIDS, is an enveloped virus. It does have a capsid. You can see the capsid that surrounds the RNA core. And then it has an envelope. And then the glycoproteins that attach to the host are on are embedded within that envelope. And reverse transcriptase is an enzyme that is also inside of the capsid. So there's RNA and there's also reverse transcriptase. And what that does, we've learned that transcription is when DNA is converted into messenger RNA. Well, if it's in reverse, the enzyme reverse transcriptase can take the RNA in the core of the HIV virion and convert it into DNA. And then your DNA can go through the processes of transcription and translation into proteins. Okay. So we know that we can classify viruses by their genetic material and the design of their capsid and whether or not they are enveloped. This is an image of the virus that causes rabies. This is an image of the virus that causes smallpox. It's called variola virus. That has been eradicated through vaccination throughout the world. And you can see in the illustration that this, up at the top left, that this particular virion has a capsid, and then it also has an envelope. And the picture on the right, same thing. It has a capsid, double-stranded DNA on the inside of that capsid. Here's the capsid. And then an envelope on the outside of phospholipids. Okay, so you don't have to memorize this list, but some examples of RNA viruses are rabies and retroviruses, like the one that causes HIV, the one that causes the common cold. DNA viruses are herpes viruses and the smallpox virus. Single-stranded RNA viruses are rabies and retroviruses. So these two examples are actually single-stranded. And then double-stranded DNA viruses are the herpes viruses and the smallpox virus. And the ones that have a linear strand of nucleic acid are rabies viruses, retroviruses, herpes, and smallpox. So all four that are listed above are linear. And then some viruses have circular DNA or RNA. The papilloma viruses, you would probably recognize that as there is a papilloma virus that causes genital warts. And bacteriophages, which infect bacteria. Non-segmented means the genome consists of a single segment. It's not broken up. The para-influenza viruses are an example of non-segmented. So maybe their genome is either a circle or it's just not broken up. But an influenza virus would be like different components of the genome broken into at least two segments. So classifying by capsid structure, the virus can be naked icosahedral. We saw the adenovirus earlier. Hepatitis A and polioviruses. And then enveloped icosahedral. The herpes simplex virus, Epstein-Barr, rubella, yellow fever, and HIV